In the previous lesson on synaptic plasticity, we learned about the basic phenomenon of long-term potentiation. This was discovered in the hippocampus in the 70s, and scientists demonstrated that the strength of a synapse could indeed change, so this could be a biological mechanism for learning and memory. We also learned the conditions under which LTP can occur, and it satisfies Donald Hebb's suggestion that neurons that fire together wire together. You must have target cell activity and presynaptic cell activity, both, for LTP to occur. If you inhibit the target cell, no LTP. If a weak synapse cannot drive the target cell, you'll get no LTP. But if you stimulate the target cell, give it some help, then you can strengthen that synapse. And if you simply stimulate the target cell by itself without stimulating the presynaptic terminal, then you'll get no LTP. So neurons that fire together wire together. In this lesson, we'll look at the same phenomenon, but with a little bit more biological detail. First, some review of basic biology. Here we have a target cell, so we got the cell body, its axon and terminal, and then dendrites. And all these dendrites are spines, and the spines are the sites of synapses, where a presynaptic terminal makes a junction with a spine. So that's called a synapse. So we have the axon terminal here. Neurotransmitters would be released into the synaptic gap. They would bind to the receptors, protein receptors on the membrane of the dendrite, and then those receptors are channels that would allow ions into the spine. So the sodium ions are shown coming into the spine here. So when neurotransmitters are released, you'll get a little bit of an electrical signal in the target cell. For each of the synapses that is currently active, some amount of sodium will accumulate in the dendrite. If we stimulate with high-frequency action potentials one pathway, we get a lot of neurotransmitters released, so a larger postsynaptic current. And we might even reach the threshold for the target cell, and when that threshold is reached, it will generate its own action potential that will race down its axon, and then neurotransmitters are released here. Now, there are two ways to activate a target cell. You can do it this way, where you repeatedly stimulate one out of a set of inputs, but you do so, the repetition, the high-frequency action potentials will release enough neurotransmitters to generate enough postsynaptic current here to reach threshold. So that's one way to activate a target cell. Another way is that you have multiple synapses active simultaneously. At each location, you have a buildup of ions until you reach threshold, and then you'll get an action potential. Okay, so let's take a look at the basics of LTP. Okay, so here was the fundamental discovery. We have our target cell, we have our presynaptic pathway here, and we're going to send a test pulse, one action potential down the axon to the terminal. We will record some amount of current here, so there will be some depolarization. The sodium will come into the spine at this location. So if we had a recording electrode in the cell, we would record the target cell's response. So each of these dots would be the data from sending one test action potential down the pathway. But notice then, if we just send one action potential down the presynaptic pathway here, we don't reach threshold here, so we're not getting an action potential in the target cell. We're simply measuring the effect that one action potential in this pathway has on the target cell, and here we see the data right here. Next, what they did was they sent the high frequency train of action potentials down here, maybe 100 hertz. Well, that, recall, is going to release a lot more neurotransmitter. You'll get a larger postsynaptic current here, so you get a larger depolarization if you're talking about voltage. So, a lot more sodium enters. We reach threshold, we get an action potential. So the high frequency stimulation of that same pathway is going to generate a target cell response. But now we go back to that same pathway. After we do this high frequency stimulation, we're going to send one action potential down it again. Here's our test pulse again. And notice, after the high frequency stimulation, a test pulse generates a larger response in the target cell. I'm showing it here with a little extra sodium coming in for each action potential that arrives at the synapse. So afterwards, another test pulse has a greater effect on the target cell. The synapse has been strengthened. This is called long-term potentiation. Notice it's long-term, it's lasting for 8 hours or more. And now, suppose we inject negative current while we're giving that high frequency stimulation to that pathway. You'll notice that if we prevent the target cell from firing its own action potential, we get no LTP at the relevant synapse. In addition, the LTP that was generated at the synapse is specific to the synapse that was given the high frequency stimulation. So for example, this synapse here that was not stimulated will not enjoy LTP, because while the target cell might be firing due to the activity along this pathway, this terminal was not active at the time the target cell fired. And you recall, this harkens back to the conditions under which LTP forms. We have to have both presynaptic and postsynaptic activity in order to form LTP at that synapse.